So that in fact also explains why Mikhail is not doing it, not here today, because he had issues with arriving in time. Just like you, in this particular case, there was more planes than subways. So he will talk next Tuesday. That's the lecture that I announced for today, but it will be next week. Okay, today we talk about protein interactions. Let's begin with the terminology of protein interactions. So this is one particular view of a bunch of proteins that interact with each other. Here, and the structure comes from the lab of Wayne Hendrickson. Here is sort of the more molecular view, and in the molecular view you may begin to appreciate that exactly here is one protein, so-called GP120 is here, this dark blue. And GP120 stands for glycoprotein 120, and that is an HIV protein. This was a very important structure determined by Wayne Hendrickson 14 years ago. It is complexed, meaning binding to antibodies here, 1, 2, and to CD4, which actually has sort of two different regions here. It's this elongated protein here. The biology of this binding here is very important. Now, just about the definition of what we are talking about when I talk about protein-protein interactions. Essentially, last semester was about forces that keep internally the protein together, make the protein fold, make the protein adopt a unique three-dimensional structure. And this time what we talk about, what is more functional, is when the protein interacts with something outside of itself, with something other than itself. And those interfaces are shown here in red, here in blue, or this may be called an internal one. So I'm talking about protein-protein interaction if those two proteins touch at some point in time and not at others. Meaning they are at some point in their creation independent proteins that have to meet. And typically when you read the literature about protein interaction, what people usually talk about is association. The type of A activates B, activates C, activates D. Then they would say that all of these here are associated. They're associated because they partake in a pathway. The D, A, B, C, D is needed if you want to set on a certain process. If you want to do something, you need all four of them. In that sense, they interact. That's very often what people talk about interaction. So for us, really, it is these two don't interact, these two do. Whenever there's touching, then we talk about interactions. There are different ways of measuring such interactions. One, again, you determine the crystal structure. You freeze them, so to speak, just like you determine three-dimensional structures of proteins in general. Not that easy to do, but there are hundreds in the PDB, in the protein data bank. Another way here is the so-called yeast-to-hybrid system. The yeast-to-hybrid system is biology usually works with markers, and this is one sort of marker. You put on one protein the binding domain, on another protein the activation domain. In the moment that these come together, something is visible, okay? That means that when these two proteins to which you attach them bind, they come together, you get a signal, okay? Yeah?
Could you explain me what do you mean for marker?
This, biology uses the word marker to indicate, but there are biomarkers, for instance, that have to do with medicine, biomarkers that define who you are, or distinguish between different people in the room here. And marker are just ways in which you put some construct that you construct, the BD, the AD here, into a system being the cell, being the two interacting proteins, and make sure that you see something that X and Y do. Okay. What you notice is the signal, just like GFP, green fluorescent protein, is a marker, because you attach it to another protein. Since it emits light, you see something about the other protein. But the GFP itself is the marker, is the thing that your tag, let's call it tag, sorry, I was looking for the wrong word. It's a tag, just imagine it to be a tag, okay?
So it's just to make it visible, not like an enzyme that helps a reaction.
Yes, exactly. So if it would help the reaction like an enzyme, then you would actually change the biology. So all you try to do here is to have something that gives you an ability to mark, to remark, to measure an interaction. And that is one way to a relatively large scale experimentally measure interactions. But still, for most human proteins, we do not know their binding partners. So many of these histio-hybrid experiments have been done. They can be done on a large scale. You can do query thousands of interactions in essentially one experiment. But they're also very faulty. Yes?
So what is the thing you measure as output then?
You essentially, the activation is just you mean the signal? Mm-hmm. I believe it's light.
Okay.
You attach that further and then you see the light. Okay, let's get back to homology-based inference. We have talked about that again and again. If I want to now homology infer protein interactions, so assume I have the interaction between A and B and I know experimentally that they interact. Assume I have another pair of proteins, A prime and B prime. My question is, at which level of similarity between A and A prime, at which level of similarity between B and B prime, can I infer that these two in fact also interact? Now, the story got more complicated with respect to what we did previously for subcellular localization. For subcellular localization, homology-based inference only needs one pair of proteins. So homology-based inference for subcellular localization means I know that this is a nuclear protein. I know that this is similar to it. Can I infer that it's subcellular localization? The story got more complicated here because I actually have two levels of similarity. And you immediately have new options. And that's a lot about the problem of computational biology. So you could ask, you could measure this such that you look at the measure whenever either of these two, so x1 and x2 here, you take whatever is the minimum, you take the average, you take the maximum. When you scale how well is something conserved, you now actually have x1 and x2, right? And how are you going to measure now similarity? And there are obviously different choices. So you can measure, you can average over the two, you can take the minimum, you can take the maximum, and all of these different choices means essentially complications. And there is no clever way in which you can settle these complications. Anyway, the story is the same as before, just got slightly more complicated. And obviously the question is, can we transfer protein interactions through homology? So do proteins in different organisms interact the same way? In some sense, the answer is very obvious. You all know that we do interaction studies in yeast, mouse, in order to probe, I've told you that, to probe human interactions. So if the answer here would be that we cannot do it at all, then all of that would be nonsense. All of that would be meaningless. Because ultimately, protein interaction is one of the crucial components of protein function. Proteins don't function without binding partners. They function in networks. They function in concert. Okay? The base unit in some sense is the interaction. That means that if you could not infer from A and B in mouse to A and B, A prime and B prime in human, there would be a problem. Okay, let's just look at how well it's conserved. This is the H-val. The H is the t distance. Remember, there is this blue line here, and this measures the distance from the line. Minus is below here, and those are all plus values that I show you. This is some measure for the degree to which it's conserved, and that the numbers don't really matter. What you see, however, is we have large error bars because we don't have a lot of data. Warm versus not warm, so this is an old paper from 8 years ago. And there are two ways in which you can read that. One is there is some drop and some up, so obviously this is the noise, but there is some function that you most likely see in here. And then there is a drop somewhere here. Clearly, so the blue line here, the 0 line, was the line at which you could infer similarity in structure. Clearly this thing drops before that. That much we can see. So somehow if protein interactions are conserved, then they are conserved for more higher levels of sequence identity. Okay? So again, this one is, I look at A and B in worm. I ask whether A prime and B prime in human are in fact also interacting for this data set for which I know the interactions. Once I convince myself that they do, I can use that to in the future predict homology-based inference, predict interactions. Now, so one thing is clear here when we look at structure or other features of function, that looks lower, these curves. And now back to the number, this is actually in principle the number that compares to that number. So there, by looking at the values here, we go to 10. So on this plot we would be here, and you see that this is lower than all the other things that were enzymatic activity, structure in green, and localization. Okay, but we can do it to some extent. Now let's just look at gene transfer. Let's look at the background idea of homology again. Homology again is the similarity of a functional unit, so the hand in human and the same in the whale. That in some sense does something slightly different. Now on the level of the genome, so we begin with a species here, we duplicate, we have a speciation event, we have essentially this similar setup of the entire genome. So in this particular case my species exists of 3 genomes because I'm lazy. And those 3 are still here, and we would call them orthologs, we would call them the corresponding identical genes. And at some point there is a duplication event here. We still have species B1, but within species B1 you suddenly have a duplication of an entire gene. Yes?
That's a duplication?
Duplication, you simply take this gene here and make another out of it. And by introducing a new symbol here or a new shape, I try to indicate that what can happen now is that this can be slightly different.
It's a mutation?
The point is at the moment of the duplication, you typically don't have a mutation, you take the entire thing. But then, because the image is that since you already have this gene and it does what it does, this is free to evolve because you don't have as many functional constraints. Because whatever you have to do with it is anyway done with this one. So you somehow gain a new copy with which you can play. And if it brings you an advantage, the advantage can be twofold. It can be the advantage from doing the same thing as that one, but you do it twice, so you're more efficient or something like that. Or the advantage could be you do something slightly different, which allows you to mutate, and you can do something new. And this, in fact, is something that we see all the time. Then at some point you get to a new species. And again, in this world of orthologs and paralogs, all of these here, these squared ones, they are orthologs in the sense that in a different species, they are the gene that do the corresponding thing, that do sort of the same thing. Okay? That's the idea. While at the duplication event here, new things came in that may do the same thing or may not do the same thing. All right? And we see this all the time in nature. Here's an extreme example of that, where the sea slug in fact incorporates chloroplast from a plant and then actually does photosynthesis for 12 months. So you eat something from a tree and you suddenly grow like a tree or something like that. Imagine this. This is typically what happens in nature, and at some point it's just eaten up and is used in a different way. On the level of the genes, we have cross transfer of genes across the trees. Mitochondria is an example of that. I told you that the mitochondria in eukaryotes in fact are a relic of a bacterial world, and that is such a cross gene transfer. Okay, so we have transfer of genes, we have duplication of genes. Now let's rephrase this issue of the homology-based inference for protein interaction in the context of different species. Now we have 2 possibilities. We can say, okay, A and B I know experimentally interact. I have a protein A prime and B, another one in B prime. They are similar, but they are all in worm. So in the level, what I showed you here, they are sort of this one here, right? And I can ask, how well do they? What's the probability for them to bind? And I can look at the same or not the same pair, at another pair that I believe is the one that corresponds. I should have been better here. So those are now the square ones here, essentially, and look whether they interact. And what I'm going to do is I'm going to look. By the way, what I showed you before is the minimum of the two values, x1 and x2. And I'm just going to look at pairs in the same x1 and minimum x1 and x2. Let's back again. This is warm versus not warm. So this is the orthodox. Those are the genes that in a different organism are supposed to do the same thing. This is the paradox. Those are the things that are duplicated, that are not constrained to do the same thing. Here's the same thing for fly versus, so this blue here is warm versus warm, so C. elegans. Here the blue is fruit fly versus fruit fly. And the green is again the fly against the rest, against other organisms. Warm against other organisms. So what do you observe?
In the same species, proteins tend to interact much more similar with the same.
So essentially what you see is that the paralogs, the ones that are not constrained evolutionary to interact, those you assume exactly do the same thing. And they are less likely to interact. That's the green ones.
But with less likely, we are still speaking between 0 and 10%. Because this is like...
But that's true what you say. So, but now the problem with these, now we get to the problem with these numbers. Part of the problem with these numbers is, how do I compile accuracy? What I do is, I take a set of protein interactions. I take, in this particular case, C. elegans, take all the known interactions in C. elegans, and say they are true. Now, where do I get my false ones from? How would you get the false ones? In order to compile accuracy, you need false ones, right? Who gives you false ones? In the same species. So you have a set. Let's just stick here. We're looking only at warm. I give you a data set of all the proteins that are currently known to interact in warm. How do you get the false positives in order to compile an accuracy level here?
In the same species.
Mm-hmm. Well, everything that is not observed is not right, and that obviously is not true. Because this, since I already told you that most interactions are not known, so this is just an estimate. Okay? And this is an underestimate of performance. So, how much of an underestimate of performance, we absolutely cannot tell. It could be true that if I read this plot, we still don't, we are nowhere near saturation even in yeast. But at some point we will be. And at that point, when we redo the plot, I have no prediction. I cannot say whether the number will change dramatically. But at least what I can say is, I cannot predict it today. So today that's a number that makes some sense, and is in fact the number I measure. But there are, in this particular case, too many assumptions in my number. So in the case of the protein subcellular localization, I have a set of proteins that I know are nuclear, a set of proteins that I know are not nuclear. So every one that enters my scale here is a measurement. In this particular case, I cannot do that. That is another problem. So for the moment you talk about protein interactions, you somehow square many problems. And that's another problem you buy into. But other than that, it's very clear, standard error or what, the blue ones here are higher. So it seems that this here is more likely to interact than that. And that brings up the issue of why. And ultimately, the answer to that question is absolutely unclear. So we posed that question in 2006 and have been running around ever since. So there are now many other publications that show something like that. But why this is the case remains a puzzle. It does, however, imply that paralogs seem to be conserved more than orthologs. And it does imply that there are issues with model organisms. What exactly that means, we don't quite know. what exactly that system is. So I believe one thing that sort of has been evolving over the last few years from others publishing that in different contexts, for instance also for Go numbers, is that it buys you an advantage to keep a protein that does the same thing around, to do the same thing twice if anything happens. For very crucial proteins, the duplication of it is actually an advantage. And ultimately that may just be the explanation, that there in fact is there are more constraints evolutionarily on paralogs than we had thought. Okay, now another story here. The story of measuring the same interaction twice. So in this particular case here I looked at homology-based inference, so I had another protein in a different organism or in the same organism that is some sequence similarity. Now I'm going to look at the same protein, A and A, and B and B. The only difference is I measure it twice. Okay? And I'm asking the interface between these two, where they touch. In these two interactions, in these two cases in which I measure, is that identical or not? Right? And so that was the project of Tobias, who in fact talked to you, and is one of those things that Tobias stumbled over when massaging the data. It never occurred to me that the answer could be that they are not identical. They must be, and if they're not, then it's a problem in the measurement. That was my strong conviction, because otherwise the reliability of that interaction has to do with the reliability of a protein. What Tobias clearly showed, and there was a publication 2 years ago in PLOS Computational Biology, is that this here means difference, this means the same. Here the interfaces are the same, and there are different ways of slicing the pie. Here is a different way of measuring similarity. Again, dissimilar, similar. Whatever these distributions, they typically all look like that. The majority of things are here. So the majority of things are in fact the same. But we always see some large minority, bless you, some large fraction of things that in fact are not similar. To show you a few examples, the green and all these color things here is a 2-on-2. And you see that the different color modes here differ very much. So whether that green thing, or this is one mode of binding here, these three, and this is another mode of binding, and they're very different, right? Here, these two are binding very differently, so completely differently, to that protein here. Here, they bind even on different sides. In this particular case, they're both blue. Same thing here. 3 different binding interfaces. So many of these are not subtle changes. They're large movements, and they seem to be biological. How can that be? How can it be that the protein functions in this configuration and in that configuration? And again, it's another puzzle that is out there. We have examples, in a few collaborations, we had examples where people see molecular reasons for that. So where this way of interacting triggers a slightly different detail than this mode of interacting. How that is determined is beyond us at this moment. Again, it's the same structures. Maybe the environmental change. Now, initially, or 16 years ago, my idea was we just take 2 proteins, protein 1, protein 2, and predict that they interact. And the way I predict this, I take a stretch here. In this particular case, I show 9 consecutive residues of the green and the magenta here. And I simply ask, are the central 3 of both in contact to each other? Okay, and I trained a neural network on that. I did that in '98. And it was by far the best failure I ever had in my life. It absolutely didn't work. That made me begin to realize I have to find ways to reduce the false positives. In fact, that was the problem. There's so many proteins that might interact if you put them in a tube, that might interact if they meet the right way, but in a cell they will not. Okay? So the prediction is wrong because the cell has other realities that I in my prediction system don't see. So here's the first way in which I sort of reduced false positives. When you predict surface residues, you can say that about half of the residues are on the surface. Now, proteins interact through their surfaces. By simply cutting out everything else, you reduce the problem by a factor of 4. You essentially have only 1/4 of the data points left. Right? If half of the data is on the surface, then you kick out 75% of all your false positives. And in order to do that, so initially we had a method that was good at predicting the center of a protein, the core, and now we needed a method that is good to predict the surface. We developed that, and by that sort of we had our first filter. Now our next filter was in fact to predict external residues. So good to know that this is the surface, but would even be better to know if I knew which residues bind. Not on both. I don't know the binding partner, but at least for one protein, I want to predict that these two residues here are going to bind another protein. Okay, and I will get back to that in a minute. And then we get into the next issue here, the subcellular localization. Say 1/5 of all the proteins are nuclear. So now we have a factor of 25, so 1/5 by 1/5. A large reduction by simply looking only at pairs of proteins in the same subcellular localization. By the way, here is in fact the image of how often do two proteins interact from that are secreted, cytoplasmic and so forth. Green means much higher, 2 standard deviations higher than expected. And you see most proteins mostly interact with proteins in the same subcellular localization. And then there are some exceptions here, but again you can take that into account. You can take this table and turn it into a probability from the prediction of subcellular localization. So for that you need a prediction of subcellular localization. We have that. Then we can predict proteins that are binding to other substrates, that are competing with protein-protein interaction, like protein DNA. We'll talk about that later. And then we can improve alignment methods. We can better predict domains, sort of the units that interact. And then we can put all together and predict protein-protein interactions. That is essentially what our group has been doing for over 10 years. I've shown this slide in many keynote talks I gave all over the world. The only thing that changed on this slide for many years was the face. I said, Tobias now, before the same person had a different name, and before it has a different name, because for years and years and years, the face on this slide just went ahead and did something else. They started working on exactly this last step, but somehow that's not what came out. This is the first time that I'm really going to show you the last step. So Tobias just, we got a major revision on the paper, and I'm going to talk about it later this year or early next year. So now we really have the first method that predicts protein interaction. But let's begin with the beginning. Let's get back to the prediction of the interaction sites. And for that we need data. Again, the first question that we ask, if I want to predict from sequence which residue binds to other proteins, my first question is, where do I get the data? The answer is I take it from the PDB. Now the first question becomes, well, I'm looking essentially at the features of binding residues, right? So the interaction sites between different proteins and the interaction sites inside of a protein, is that the same biophysics? If it would be the same biophysics, if it would be sort of the same environment, the same rules, then I have a huge data set from which I can learn. I take the entire PDB, I have 100,000 proteins, I can redundancy reduce and get a large data set of contacts between residues. If, however, I really need to take the relatively few hundreds of structures for which I have these complexes, these interacting proteins, my data set will be smaller. So the first question really is, do these interfaces here between two different proteins, inside of a protein, and here between, how many am I going to show? Oh, between homomers, homodimers, between different domains. So there are different ways in which I can slice my data and ask, Do they differ? Are they the same? What you will look at now are amino acid compositions. Okay? So, here's the data. I have 6 different types of interactions. Intradomain, the yellow one, is just inside of a protein. And the blue one here is what you would call the typical transient protein-protein interaction. I have these 20 amino acids. I have a log-odd scale that measures the surprise. So the surprise, essentially, the background is all the occurrence of arginine, lysine. So these are the positively charged, negatively charged residues, hydrophobic residues. So they're grouped by different, somehow different features. Now, below 0 means less often than expected from background, from this entire sequence. More than expected in that class above 0. Do they differ? Let's simplify this here, and let's only have 3 states. In the 3 states, and I'm sorry I changed the coloring here, internal, inside of a protein is green. External protein is, in this particular case, blue. We show the same thing. So, do they differ? Is the physics, the biophysics of external binding the same as internal binding? It's not, you say. Argue. Can you argue? Or can somebody else argue? Whoever wants to argue. So the statement, please, you have to argue why it's not the same. Shouldn't be too difficult, right? So shoot. You're the only one who is moving. I don't know why. Huh? No, but that's not what I wanted. I want everybody to. What are the observations? Can you see? Can somebody please describe the plot?
Describe is too much. I could say for me that from a hard feeling of this data, I see too much difference with external. I don't know, I think so.
So when you present your results in your paper, this is the figure. Begin by making observations. So every reader can see that figure. But you guide the reader, and the first reader will be a supervisor. You guide the reader by, in fact, observing what you believe are the most important aspects of this result. What are those? Just give me some. I believe the first result that I clearly heard from you is that the bars differ. Almost everywhere, blue, green, and red differ. That's the first statement. Is there any other statement that you want to make?
For the same amino acid, some bar have positive value and some bar has negative value.
So the degree of difference is different, varies with the amino acids. So they don't uniquely differ. And when you look, let's just back you up on a feature that you're not necessarily supposed to remember, but these R and K are both positively charged residues. And you do see that, in fact, clearly this one here has a very different behavior from the negatively charged residues, and a slightly different behavior from almost everything else, but remarkably, the 2 positively charged residues have a different behavior. Again, it's not only that the 3 colors differ, not only that the residues, as you said, differ, but it's even essentially not the same within a class of things that you would say behave similar according to their biophysical features. Those are 2 negatively charged residues, and they sort of behave similarly. But they absolutely don't. So they do what we expect, they don't. And you see a similar feature in other groups here. Okay, anything else that you see? Remember, the 0 line is the line of expectation.
The third group is way below expectation, more than the other ones.
Yes, that's another important. So, which is sort of a negative statement, right? the protein interactions are defined by absence of expected contacts. It's not quite true, so we have some exceptions to that, but it's true. It's an interesting finding. Yes. Anything else? For me, something that jumps at me, just think about all three together as a group, as one group. Just look at the zero line. It all differs from zero, right? Wherever you go, whichever bar you essentially look at, there's almost not a single amino acid where not all three, again, there are some issues, but almost always they differ from expectation. What does that mean? How can that be? So typically, when you normalize, so when you essentially, the three different types of classes that you present here are supposed to somehow reflect all situations. When you reflect all situations that you measure by background and everything differs from zero, this is the moment when a flag should shoot up and check my code. This sort of smells like a bug. Usually, if you have an exhaustive set of parameters, it cannot be. In this particular case, you don't have an exhaustive set of parameters. In fact, they are all selected for binding. What enters this plot are all the pairs of residues that contact in different environments. But what we immediately see is that whether or not you contact is clearly imprinted upon the amino acid sequence. It's not chance. The number of arginines that you see in contacts is nowhere near what you expect. And none of these three binding interfaces, right? That's an interesting observation in its own. But there's another point here. And that other point essentially is the question, is it meaningful? Is that a meaningful observation? And what is it that you would do in order to answer that question? So the question really is meaningful. Look at this one here. All we said is true. The three colors differ from zero. The three colors essentially differ from each other. But the difference is small. Here it's much larger. And there are many examples here. So there are more extreme examples. Let's just look at the blue that you don't see here. The isoleucine. Is that important? Is that meaningful? How would you figure it out? So how would you figure it out? That's of course the first question you would address, right? Is it statistically significant? How would you do that?
Repeating experiments over and over and over again.
How? How do you test statistical significance of a data set? Of a difference that you observe? Anybody in the room who could give me a test for that? How do you measure statistical significance? Heard about chi-square test? Okay, so chi-square is typically what is employed in order to measure the difference in sample points. But chi-square in this particular case doesn't work. Here, in fact, the story that is raised here is that the chi-square gave very, very significant differences, and they were, because of a variety of reasons, not meaningful. Here's a different way of doing it. This one, we call it find self. So you take your data set 1, say the internal interactions, data set 2, the external interactions, and data set 3, the homodimers or something like that. Now you take 1,000 of the amino acids out here, 1,000 here, 1,000 here. And I take another 1,000 of this one. And by some similarity measure, in this particular case is the Jensen-Shannon information content, you simply ask, this one here, the last 1,000 you picked, is most similar to that, most similar to that, or most similar to that. And then you repeat this test. Essentially take out a bunch. So in each of these pots in the experiment that we have, you have about 1 million data points. You take 1,000 out and ask, what is this 1,000 most similar to? And then you repeat that 1,000 times. And this is what you get. So again, we differentiate between different types of interfaces. This is inside and this is outside. This is essentially what we want to look at. And the first thing you observe is there's always the highest number in the diagonal. And that essentially tells you that on a large set, you can distinguish between them. That tells you that these interfaces really differ. Okay? Now, if they differ, we can develop a prediction method. And how would you do that? We would start with a data set from protein interactions. I'm sorry, I don't understand why this was supposed to be out. That's the way you construct the data set. So now we want to have a prediction method. So we know that for a set of 1,000 I can distinguish between external residues that bind externally and residues that bind internally. But now my task is to predict the binding for a particular residue. And one thing that I have to establish is that my binding interfaces are consecutive in sequence if I want to run a sliding window. Consecutive in sequence, here's one particular way of looking at consecutive in sequence. That's an important thing to remember when you think about windows. So typically consecutive in sequence, you could imagine that you have a running average. Or you could look at how many in the window are in a binding interface. Now, if you had a binding interface that looks like this here, so this is the protein it binds to, and that's the protein that is binding. You see that sequence consecutive, these are all binding here, but they're all not sequence consecutive. Essentially, it's like fingers binding to this, but the thread goes like here. So if I just cut out a window of 6 consecutive residues, 1, 2, 3, 4, 5, 6 or something like that. In this particular case, there would be only one in the binding interface. And for all of these, it would be a little bit like that. Now, in a little bit less extreme example, it could still be true that, so let me just have it like this one here. So again, that is my protein it binds to, and now my binding is like this here, and this is 1, 2, 3, 4, 5, 6, 7. So now, essentially, I would see that there is some consecutive signal, but the signal is not really consecutive in terms of residue by residue. So this one and this one are off, this one is on. But in a window of 9, you have 3. And this is the way I'm looking at it. So I'm looking at a window of 9, and I'm asking how many of these 9 residues are in some of these interfaces. And there are different decisions about what an interface is. The yellow one is tighter, so they're really much closer. It's 4 angstrom. The other one is 6 angstrom, whatever that means. You don't need to remember. But essentially you see that the distribution is such that it's certainly shifted toward this end. The tighter you look, the more the distribution moves. The yellow one is more in that direction than the blue one. But both of them have averages that are higher than 1. Okay? It's not that in this first example I showed you with a finger that every residue binds, but there's nothing in the 9 residue environment that binds 2. That is not the case. Clearly, the distribution of all the interface residues is such that they're in the vicinity of 9. There are more than 1. In fact, again, you may say it's 5 here for the yellow. You may say it's 6. But clearly, it's a collective phenomenon. So cutting out a window of 9, you clearly see a relatively strong signal for protein interaction in there. So that is what we can use for machine learning. When we apply machine learning, the first question is, how do we choose the input features? So how do we? And let's just have that as a discussion between you guys. Can we sort of somehow form groups of 2? Homology or small subgroups? And you come up with ideas how to do it. Yeah, you have to find your way. But you did find your way.
Can we hear the question statement again?
So what we want to machine learn that a particular residue is on the surface and binds to another protein. So I give you a data set. What is your input for your sliding window? I can also tell you that in this particular case, when we started this, we had in the ballpark of a ballpark of 50,000 samples. Positive. We had millions of negative, but positive we had 50,000.
With positive mean
Binding.
Binding size.
Mm-hmm. How do you decide on the input features? Nobody has an idea? So let's do it together. Better. Okay, so what can we do? How do you start?
I would just take the residues.
So we have, what I showed you before, remember, is the, is this one here. So in a window of 9 consecutive residues, you clearly have a signal. So my first question to you, if you run a sliding window, what is the size of the sliding window? It's going to be less than 9, more than 9?
You said that it was 500? No.
So we have about 50K positives. So we have over 100,000 examples. Because the negatives far outnumber the positives. Total we have about 100,000.
So if you have a signal with a sliding window size of 9, I would take 9. Because you know there's a signal. So I would take a feature vector with 9 inputs. Each of them is coding one residue. And the next feature vector is just the sliding window shifted from position in the whole data set, and then I would feed it into the neural network like this.
So residue 1, 2, 3, 4. I take 9, right? But you guys know I'm lazy, so let's redefine 9 to be 3. So a window of 3. So my first window is from residue 1, 2, and 3, right? My second window is?
From 2, 3, and 4.
Okay. And then your output codes for what?
Binding or not binding?
So the entire thing.
Yes. So that if there's a signal inside the sliding window.
So first of all, I have to admit that we never did that. So I'm not sure how that would work, and I have reasons why we didn't do that, which essentially are only historical. So the reason why we historically didn't do it. But it's also true that if the student who did it, Janai Orfan, who published this paper here, had come to me and had proposed that, I would have said, okay, if you at the end of the day your method succeeded, then your method would be able to, on a level of 9 consecutive residue, tell me one of these 9s is binding. I'm not content with that. I really want to know which one. So for me, if I believe I can do it, I would actually believe I can distinguish between the one that binds and the one that doesn't bind. Not only between the 9-mer that has more binding residues in it than the other 9-mer that has fewer binding residues.
But given the feature vectors, if it is a sliding window with 9, then I can combine the information anyway from those 9s and select the intersection. And then I also get a more distant, more precise solution.
No. Well, that actually may not be the case. Given the observation here of the distribution, that may not be the case. So if you average, so you just look at this plot, move it one further. And then you average over a bunch of, let's call it Gaussian. Okay? Averaging over a bunch of Gaussian gives you another Gaussian. It does not give you a mountain. Don't forget that. So you have this Gaussian and that Gaussian. Together they give this Gaussian, not a mountain. So that means from averaging over different ones, you do not get the cut and then you say, okay, that left is my residue or something like that. So the procedure that you're talking about to, in fact, then run this energy landscape of 9-mers and thereby find the one that is more likely is not that easy.
Yeah. That's true.
So you build in a noise in some sense, and you build in a new problem. So I don't find this a good idea. I really would argue in the case of 3, so this is what we did, and this is what I would argue is better, that the 3-mer, 9-mer, right, is coding for the central residue is in a protein interaction or not. Okay? And then you move it ahead. Now, the only issue here is, let's talk about how do you code amino acids. How many input units in this idea of your 9-mer window? How many input units is one 9-mer window? No matter what the output coding is for. But the input, how do you, in terms of an SVM or neural network, or how is the input feature? How do you code the sequence?
We have a detector with the size of all possible amino acids, 20.
Okay, 20, it's 20. So in this particular case it would be 9 times 20. Right? Everybody okay with that? Can you think about any other way of doing it? You can, just give me alternatives. This is one solution. And it's the so-called the sparse coding. The sparse coding, because in this particular solution, you essentially, again, I'm your lazy teacher, so let's assume we have only 5 amino acids, then this vector for each of these 9 would look like one of these, right? And I'm going to be even more lazy. The rest you do by extension. So 1, 1, or 9 ones, and 9 times 19 zeros. Right? That's a very sparse coding. There's a lot of 0 in my vector. Is there any other way you can imagine to do it? Sure. You could code them by you could attach features. Remember, we had positive charge, negative charge, hydrophobicity, or size. So you could imagine that you have 5 or 6 different features, and you put the amino acids in a grid in which they all come. And then rather than a 20 vector base for each amino acid, you have a 5 vector base. The problem, it turns out that you could do many, many ways, and they all don't work as well. And there's a longer argument why. So the sparse coding, that is in many ways the dumbest thing to do, is the best thing to do because it makes the fewest assumptions. All the other vectors that I would construct, in what I just proposed, they make assumptions about correlations. This one doesn't. Okay? Or it does, but it's the least possible assumption it can make. Anyway, now here's another issue with that. So if that is my first residue, second, third. So say my window W is only 3 because I'm lazy. So for W of 3, the first central residue I have is number 2 in a protein. My window is 3 residues long. I code for what is the state or what's the binding state of the central one. So the first one cannot have a binding state. That is my first window. How can I predict for the first residue?
That's why I said 21. Maybe Rost, you can also put a...
Oh, 21.
Some edge.
Oh, you said 21. Oh, sorry. I'm sorry, I misunderstood why you said 21. Did everybody else understand why this is 21? So, essentially, you all know there are 20 amino acids. David just introduced a sort of pseudo-amino acid, a spacer. And if the central one is here, then everything before it gets a spacer unit, essentially being outside of the sequence. And this way, you can, in a sliding window approach where the central window has some feature, you can span over the ends of the proteins on either end. Right? This is the first one in a 9 residue window, so the first residue would have 4 spacer units here in front, and the same at the other end of the protein. Okay? Now we solved that. So we have a sliding window, we have 9 times 21 input units. And 9 times 21 input units, say, let's just look at the example of a neural network. Say we need 20 units. So this is in the ballpark of 200. We have 20 input units. How many free parameters essentially we have? 4,000, right? We have 100,000 data points. Is that okay? $4,000 free parameters, 100,000 data points. Okay, let's vote. Yes, no. Hand. Yes. 1. Hey, every hand has to go up for either of the two. Abstain is not an option. So, from the count I see it's 1, 1, and 2, 5, no. Why? Why do you feel that 100,000 data points is not enough for it's a factor of 25 for each free parameter? That sounds like a lot. In most examples of machine learning, you don't have that much. Sounds at least, in fact it sounds so good. Typically, rule of thumb is times 10 the free parameters. So as long as you can even fill 10,000 free parameters. So we have at least a factor of 2 or more of what we could do. Which brings me to the next issue. That means we can put more features in there that we believe may biophysically be important. What could that be? So this, we have some input features here, and then some others. Today we discussed some of the features that might be very handy here. We want to predict.
Why should we put more?
Well, so the first answer is because we can.
Okay.
The second answer is that if you train machine learning, the input space, or what you put into the input, defines how well you can solve a problem. You try to put as much meaningful information into the input as you can. Meaningful means it is the kind of information that makes the machine learning device distinguish one set of data from the other. So distinguish between interaction sites and non-interaction sites. What could be important? Clearly we know it's in the sequence, right? That's why we begin with a sequence. If, again, we have enough data points to fill our system, we should, and if we have had more informative features, then we should throw them at this system. What other features could you think about?
You already showed the plot with the different biophysical features like hydrophobicity and charge, and we can all use them and put them in.
The interesting things, so here in this particular case, maybe you have 5 different features. So this adds for the overall fraction. So I said we have a factor of 2. If we added 5 times 9 input features, that would add only 25%. So only a fourth of what we already have. So essentially we would add 1,000. So we have 5,000 free parameters. We are still far away from what we can do, provided we can learn it with 20 million. So I'm just giving you ballpark numbers here. But what else could you think about? That's true, and in fact they help. Which part of the protein is interacting to another protein? The surface, right? Why not put the surface in? How do you get the surface of a protein? Well, they would predict it. Remember I said that we developed a method that predicts surfaces very well. And that can be an input here. So now you could imagine that you're in an environment where you put in various important aspects. But here's another aspect that is more for those who attended my last lecture on structure. Evolutionary information. So residues that are conserved are more likely to be relevant for binding than residues that are not conserved in the family. So when we look at an alignment, I'm not entirely sure that I'm going to bring an alignment. This is just the idea of using that. No, I don't show an alignment. But when we look at an alignment, then there are regions that are conserved. Those regions are more important. That signal should come in here. And in fact, that is easy to get in. So instead of using sparse vectors, you now have exactly real values of conservation. And you're not even blowing up the input space for this particular case, and you bring in some important information. And there are other things. Now, we train all of this in one device that predicts whether the central residue is in the center or not. And then there are 2 different types of predictions. Here, the center one in a surface is this unit. The center one not in the binding phase is this unit. And you see that these 2 outputs both predict that the central residue is binding. But there's a difference between them. This is strong and this is weak. So in both cases the decision threshold, say 0.5, or this one is higher than this number, in both cases the same. But here the decision is much stronger. This is a reality in machine learning that you should always use and calibrate to work for you, because it provides some strength of the prediction. That strength of the prediction you can turn into a reliability index that allows you to predict how accurate you are. So now you have a prediction method, you have trained it, you have trained your system, you have some system that can predict from the training data. You do a cross-validation. So you have to somehow hide some proteins under the table here, and after the cross-validation, provided this is done in a way where you sort of begin with a sequence unique subset of the PDB, provided you know that random splits are not enough, and you avoid overlap between training and cross-training, and you have cross-training versus testing, is that all we need to do? And to some extent, yes. Now we are ready to publish a result. What this one shows here is correctly predicted by observed. This one shows correctly predicted by predicted. And when we only use sequence information, so this is the original 9 times 21 David proposed, that's what we get, the light blue. Now here's one other thing that sort of is missing, and I'm not entirely sure whether I put it in here. So there's one other thing that is missing here. Is that good or not? What do I have to also look at?
In the background?
I have the random You already saw it, or? And I'm not entirely sure why this curve. No, I'm changing the scale here. So it's the same curve here. I'm just blowing up the scale. So that's random, and that's the data. So, do we have a prediction method? Nobody has ever done that. You worked that method out. So when Yanai in 2003 did that, nobody had ever believed that this could be done. Nobody really had ever published a method. Now we have this method, and we saw this is random. This is what we have. And the question is that good? You cannot compare yourself to anybody else when you're the first. What do you do? We begin to observe the plot. Again, whenever you have results, try to write down your own observation of your results. Communicate them first to yourself, then to your supervisor, and then see that you get it in a paper. There is a regime here. This is very low. We are really talking about a tiny fraction of all the data points. But for that tiny fraction here, the blue is much, much, much higher than random. So there is a regime. Maybe for not many residues, but for those few residues, you can actually predict them. That brings us back to this issue here, because you can simply look at the strongest predictions. The strongest predictions come out here, and you can say, well, I will cut off here, I will cut off here, or somewhere there. You don't quite know for the next prediction how the accuracy will be, but for the ones you know, you can calibrate where you expect for a larger data set the accuracy to be. And for those, when you cut off, you know that you will be at least twice as high as random. At the end of the day, you have to next ask yourself, what does that correspond to? Those are small numbers here. In terms of a protein, does it actually mean that I hit one? Does it mean I predict at least one residue on each protein? And it turns out that somewhere this level here is such that in almost every single protein we predict a residue, one. Here's an example. We look at it. And this particular example, actually all the examples that we looked at, the ones we predicted, they made biological sense. Either they were correct, or they were somehow near a binding site, they made a binding site. So we convinced ourselves we have something that really is interesting enough, and for at least one residue in a protein, we get a fairly high level of accuracy. And we published that. Now, essentially, we went on to refine, and the refinement was done through the clustering that comes from here, and in fact by bringing in all kinds of additional information that uses alignments and the like, and we got the dark blue. So with the dark blue, random again is down here. That's the original light blue, that's only sequence, that is using other important input features, and you see that now we clearly outperform random, and we do that over a larger realm. So we now have sort of 50% of the residues covered. And I believe with this one, I stop it for today. Thanks for your attention.